Okay, so it is my pleasure to introduce Dr. Insup Choi, an outstanding neuroscientist and assistant professor in the Department of Pathology at Northwestern University School of Medicine. Dr. Choi earned his PhD degree in neuroscience from Aju University in South Korea in 2014. Then he pursued postdoctoral training at SUNY Buffalo and the AKN School of Medicine at Mount Sinai, where he advanced to the roles of instructor and later research track assistant professor in neuroscience. In 2024, Dr. Choi joined Northwestern University as a tenure track faculty member. Dr. Choi's research focuses on understanding the role of glial cells, particularly microglia, in neurodegenerative diseases such as Parkinson's and Alzheimer's disease. His innovative work on autophagy mechanisms in neuroinflammation and neurodegeneration has been published in high-impact journals, including Nature Communications and Science Advances. In addition to that, he is the recipient of the NIHK01 award. Today, Dr. Choi will present his talk titled Role of Microglia Autophagy in Neurodegenerative Diseases, sharing insights from his extensive expertise and groundbreaking research. Please join me in welcome Dr. Insup Choi.
Hello, everyone. Thanks for giving me this wonderful opportunity, and I really appreciate this opportunity. So I will talk about the roles of microglia autophage in a neurodegenerative disease. So since I just started my lab from September, so most of talk today will be the project that I did during the postdoc training, and then I will briefly talk about the little bit ongoing project. So first talk is the roles of microglia autophage in Parkinson's disease. So Parkinson's disease is the second most common neurodegenerative disease, nearly affect 1 million people in the US. So hallmarks of a PD is the loss of dopamine neuron in substantia nigra pars compacta. So as we can see here, the pigmented dopamine neuron are robustly degenerated in a PD case compared to the controls. So due to the importance of this neuron in controlling the movement, PD patients show the motor-related symptoms, such as tremor, bradykinesia, rigidity, and gait and balance problem. Also, PD patients show the non-motor-related symptoms, such as constipation, loss of smell, REM sleep disorder, and cognitive problem. Histological studies show the presence of Lewy body, which is alpha-synuclein aggregate, in the PD brain. In early 2003, Brock's group found out the alpha-synuclein pathology spread from subcortical to the cortical area as disease progressed. A similar observation was made in a postmortem PD patient who received the transplantation for therapeutic purpose. So it is expected to see the synuclein pathology in the host tissue, but also it was found in a transplanted tissue, indicating the spread of alpha-synuclein pathology from host to the transplanted tissue. Consistent with this finding, the synuclein has been found in body fluid such as plasma, CSF, and brain interstitial fluid. And also secretion of synuclein can be controlled by neuroactivity, and also it can be increased under stressful condition. So these observations raise the important question whether uptake and clearance of extracellular alpha-synuclein may be important to prevent the propagation of alpha-synuclein aggregates and disease progression. I'm particularly interested in the microglia, brain-resident macrophage. The microglia derived from the yolk sac during the developmental stage, and microglia precursor cell infiltrate into the brain and differentiate into the mature microglia. Microglia constantly scan the brain environment, such as neuroactivity, and also clear up the cell debris, apoptotic neurons, and protein aggregates. and they play a role in a synapse pruning process. So we ask whether microglia can control the neuron release alpha-synuclein. For this, we adopt two different Parkinson disease animal model that express the human alpha-synuclein in neuron-specific manner. The first model, we use the AAV model that express either GFP or human alpha-synuclein under synapsin promoter, which is neuron-specific promoter. After injecting this AAV into the substantia nigra, we confirm the GFP signal highly localized within UN and TH dopamine neuronal marker, but not glial markers such as GFAP or IBA1. Consistent with the others report, AAV alpha-synuclein injection caused the robust dopamine neuron degeneration. From this animal model, we isolated the microglia through flow cytometry, and we ran the Western blotting and found out the human alpha-synuclein signal in the microglia lysate. Second model, we used the transgenic model that expressed the human alpha-synuclein under DAI1 promoter. And due to the mild expression of a human alpha-synuclein, this model doesn't show robust degeneration phenotype. From this model, we again the isolated microglia and found out the human alpha-synuclein signal in the microglia lysase. So these data indicate the microglia indeed engulf neuron release alpha-synuclein in vivo. My previous lab has investigated the roles of autophagy in neurodegeneration. And autophagy is the well-known cellular degradation pathway that clear up the damaged organelle, protein aggregate, and bacteria. Autophagy machinery recognize their target and surround it through making autophagosome and further degraded it through fusion with the lysosome. LC3-2 is the lipidate LC3 form that is major component of autophagosome. And when autophagy machinery recognize their target, the selective autophagy receptors are required. So P62 is the well-known selective autophagy receptor, and P62 mediate the interaction between autophagy machinery and ubiquitinated target. And importantly, autophagy has been linked to the many different functions of immune cells, such as cytokine production, antigen presentation, and phagocytosis. And later, I manipulate the HG7 or HG14, like two different autophagy essential genes to test the microglial autophagy roles. To examine whether autophagy machinery can recognize the internalized alpha-synuclein, we inject the AAV synuclein to the GFP-LC3 mice, which is autophagosome reporter line. And we found out the GFP-LC3 structure are interacting with the human alpha-synuclein signal inside of the microglia. Additionally, we also confirmed the interaction between synuclein and P62 and synuclein and ubiquitin. Next, we treated the recombinant human alpha-synuclein protein to the cultured wild-type microglia and confirmed the similar interaction between GFP-LC3 and synuclein, and P62 and synuclein, and ubiquitin and synuclein. Furthermore, through immunoprecipitation assay using synuclein AAV injected brain or synuclein transgenic brain or cultured microglia treated with the recombinant human alpha-synuclein, we confirm the interaction between synuclein and P62. So these data indicate the autophagy machinery can interact with the ingested alpha-synuclein and microglia. To test the roles of autophagy in this context, we established the microglia-specific autophagy deficient mice. For in vitro model, we used the CX3CR Cree line and cross with the HG7 flux or HG14 flux line. And after cultured microglia from these mice, we confirmed the deletion of a target genes, HG7, HG14, and also reduced the expression of LC3-2, which is a functional autophagosome marker. And I introduced the P62 as a selective autophagy receptor, but P62 is an autophagy substrate also. So in the absence of an autophagy, P62 robustly accumulated. In vivo purpose, we apply the more specific Cree line, which is a tamoxifen inducible, and we cross with the HG7 flux line. After injecting tamoxifen, we confirmed the deletion of HG7 and also accumulation of P62. And up to 7 months after tamoxifen point, we couldn't see any difference of microglial cell number. This validation was important because a previous report showed the deletion of autophagy in neuron caused the neurodegeneration. So this data indicate the autophagy does not affect the microglial cell survival. So after this basic validation, we ask next question whether autophagy can degrade ingest alpha-synuclein. In cultured microglia, we treated the recombinant human alpha-synuclein. And interestingly, we found that the two different portions of a synuclein. The first portion of a synuclein was highly localized with the ubiquitin, and then the second portion was not localized with the ubiquitin. And interestingly, at the 24-hour point, we found out this synuclein ubiquitin double positive puncta structure are degraded. So since we already confirmed the interaction with the interaction between synuclein and LC3, synuclein and P62, and synuclein ubiquitin structure, we speculate this reduction is autophagy degradation. To confirm this, we treated the human alpha-synuclein protein to the HG7 and HG14 condition knockout microglia, and we found that the degradation of this double positive structure are blocked. And also in the presence of autophagy inhibitor, such as SIR405, or two different lysozyme inhibitor, so those degradation was blocked. And inversely, the taurine, which is an mTOR inhibitor that promote the autophagy process, facilitate the degradation of those synuclein ubiquitin double positive puncta. So next, we wanted to see it in vivo microglia. For this, after establishing the HG7 conditional knockout microglia model, and we treated the AAV synuclein, or we crossed this HG7 conditional knockout mice with the synuclein transgenic line. In both models, we found that the P62 and alpha-synuclein double positive structure are accumulated without degradation. So these data indicate the autophagy indeed degrade the ingest alpha-synuclein both in vitro and in vivo. Next, we asked the alpha-synuclein homeostasis in the whole brain level. For this, we collect the brain from HG7 and wild type, and after injecting AAV or after we cross with the synuclein transgenic line, and we further fractionated into the detergent soluble fraction and detergent insoluble fraction. And we found out the detergent insoluble fraction of alpha-synuclein were both increased in two different models that indicated of an increase of aggregate alpha-synuclein. To test it, to visualize this, we stain the brain slice with the phospho-alpha-synuclein, which usually labeled the aggregate alpha-synuclein. And interestingly, we found out the phospho-alpha-synuclein structure are more accumulated in the lateral side of substantia nigra pars compacta. And also, we also observed that this increased the signal in the striatum area. So this data show the microglia autophagy prevent accumulation of aggregate alpha-synuclein. Lastly, we asked the important question, which is dopamine neuron degeneration. And interestingly, when we delete out the HG7 from microglia, we couldn't see that much difference of dopamine neuron number. And also, as I introduced in the transgenic synuclein line, due to the mild expression of alpha-synuclein, it doesn't have any degeneration. But when we combine these two models, HG7 microglia specific autophagy deficiency with the mild synuclein overexpression and model, we started to see the degeneration of dopamine neuron. So these data indicate the microglia autophagy prevent dopamine neuron degeneration. So due to the limited time, I will just briefly explain the molecular mechanism. And when we treated the synuclein into the cultured primary microglia, and we found out that they predominantly activate the TRF4 system and NF-kappa B pathway over TRR2. And interestingly, this NF-kappa B pathway can turn on the transcript of P62. And this increased P62 interact with the internalized alpha-synuclein and associated with the autophagosome and degradate through the lysosome fusion. But in the absence of autophage, the P62 is massively accumulated and just still interact with the alpha-synuclein, but they stay there without degradation. So from this study, we show the microglia actively engulf the neuron release alpha-synuclein, and microglia degraded the ingested alpha-synuclein through autophage machinery. And we found that microglia autophage keep alpha-synuclein homeostasis and prevent dopamine neuron degeneration. So second subject is the role in a similar subject, but in Alzheimer's disease context. So Alzheimer's disease, AD, is the most common neurodegenerative disease that nearly affects 6 million people in the US. And as we know, this is the primary cause of dementia. So AD is histologically characterized by two hallmarks: amyloid plaque formation at the extracellular space, and so neurofibrillary tangle, which is tau aggregate from the inside of the neuron. And many genetic and GWAS studies have shown the microglia highly, even selectively expressed many AD linked genes, indicating the key roles of microglia in AD pathogenesis. So this is the brain slice from one of the AD animal model, which is 5xFAD. And 5xFAD robustly recapitulate the amyloid plaque pathology observed in human AD brain. And microglia stain with the IBM1 known to surround this amyloid plaque. And during this process, they increase the genes associated with the lysosome, phagocytosis, ribosome, mitochondria, and proliferation. And they convert it into the specialized type of microglia called the disease-associated microglia, positive with the CLAC7A. So we're curious about the autophagy activity in this disease-associated microglia, DAM microglia population. For this, we prepared the microglia enriched the fraction from 5xFAD brain and also limited control and ran the Western blotting. And we found out the strong increase of LC3-2 level and also the phospho 555 side of ARC1, which signaled the autophagy activation. And to visualize this, we crossed the 5xFAD mice with the GFP-LC3, again, this autophagosome reporter line. And we found that the increase of a GFP-LC3 structure around the amyloid plaque, and particularly inside of the microglia. To specifically ask the autophagy activity in this DAM population, we collect the CD11b, CD45 microglia directly from 5xFAD brain and further divide into the CLAC7A negative and CLAC7A positive cell. And when we stained this cell with the LC3, which is an autophagosome marker, we found that the LC3 positive structure are more increased in the CLAC7A positive microglia. And also through the electron microscope analysis, we found that the double membrane vesicle that associated with the other organelle, which is a key EM feature of autophagosome, overall increased in a CLAC7A positive microglia. So these data indicate the autophagy activity is increased in DAM population. To test the roles of microglia autophagy in this AD context, we established the microglia-specific autophagy division mice in the background of 5xFAD by using the 3 different line of mice, making ATG7 condition knockout in a 5xFAD. Firstly, we look at the amyloid plaque pathology. So after injecting tamoxifen, we observed the amyloid plaque pathology at the 6 months after tamoxifen, which is 8-month-old point. We stained the brain slice with the X34 dye, which is amyloid plaque specific dye. In a lower mag observation, we couldn't see any difference of number of amyloid plaque. But when we look at closely, we found that amyloid plaque become more diffuse and spiky, as indicated by the reduction of circularity of the plaque. And it's known the diffuse plaque often associated with the increased neurotoxicity. So we stained the LAMP1, well-known dystrophic neurite marker around amyloid plaque. And we found that the LAMP1 positive dystrophic neurite marker overall increased in the absence of microglial autophagy. We also directly measure the amyloid beta, which is the core component of amyloid plaque. For this, we collect the 5xFAD and Litterman wild-type control and fractionate it into the detergent soluble and insoluble fraction. And in detergent soluble fraction, we found that the monomer and dimer and trimeric size of A beta. And when we applied this assay to our interest genotype, we found that the trimeric size of amyloid beta is much accumulated in the absence of microglial autophagy. We also checked the synapse homeostasis by quantifying the level of presynaptic markers. And among the many different markers, we found that in the 5xFAD brain, there is a reduction of a VGLUT2 level. And interestingly, in the ATG7 condition knockout combined with the 5xFAD, we observed the further reduction of VGLUT2 level. But just deleting out the ATG7 from microglia didn't affect that much, which suggests that there is a synergistic effect between the microglial autophagy deficiency and amyloid plaque pathology in reducing the VGLUT2 level. So we're curious about what happened to VGLUT2 positive presynaptic terminal. For this, we stain the brain slice again with the VGLUT2 and then amyloid plaque and dystrophin neurite marker. And we can see the small puncture of VGLUT2, which is supposed to be the physiological synapse. But as close to the amyloid plaque, we found out the enlarged structure of VGLUT2 was observed. And even it was not overlapped with the LAMP1 positive dystrophin neurite marker. And when we applied this in our interest genotype, we found out this enlarged VGLUT2 structure are more prominent in the absence of microglial autophagy. So these data indicate the loss of microglial autophagy worsens amyloid plaque-mediated neurotoxicity. So microglia are known to surround amyloid plaque and is known to limit the diffusion of amyloid plaque and minimize the neurotoxicity. So we speculate microglial behavior might be changed in the absence of autophagy. Expectedly, we found that the microglia number that associated with amyloid plaque was reduced. And also microglia, the positive with the Ki67, which is cell proliferation marker, also reduced. And interestingly, the CLAC7A staining intensity was a lot compromised in the autophagy deficient microglia. To examine what happened in this brain, we ran the Baruch-RNA assay after collecting the brain from this ATG7 condition knockout 5xFAD. And we found out that most of DEG are highly microglia specific. And majority of the DEG was downregulated in the absence of microglial autophagy. And when we run the GOTAM analysis, we found out many immune activation related GOTAM was significantly enriched in the downregulated genes. And to specifically ask the DAM signature, we utilized the previous paper from the Oleg Budovsky lab. And they profiled the gene signature in the CLAC7A positive and CLAC7A negative microglia. So when we used this CLAC7A positive gene signature in our gene set, we found that those DAM signature were found in all downregulated gene. So these data overall indicate the loss of autophagy dampens the proper microglia response to amyloid plaque. So during searching the underlying mechanism, we found out a strong connection between autophagy and aging or cellular senescence phenotype. Autophagy declines with aging, and stimulation of autophagy can extend the longevity and prevent age-related phenotype. And directly, the loss of autophagy can cause cellular senescence. So let me briefly introduce about the cellular senescence. So when cells are exposed to the stressful conditions such as DNA damage, oxidative stress, or telomere shortening after multiple proliferation or oncogene overexpression, cell becomes senescent. So key feature of a senescent cell is the cell cycle arrest by expressing the cell cycle inhibitors such as P16 and P21. They are also known to secrete out the pro-inflammatory factor, chemokine, cytokine, protease, and collectively called as a senescence-associated secretory phenotype. Also, senescent cell known to exhibit the higher senescence-associated beta-galactosidase activity. When I searched this senescence and microglia terms, and I found out I could find the many like dystrophic senescent microglia terminology, especially related to the human AD. And this is one of the photo of the dystrophic microglia, and this finding is majorly driven by the Wolfgang Straight, neuropathologist. In one of the review, he drew the schematic like drawing how the homeostatic microglia become dystrophic. So highly ramified, this microglia shows some abnormal morphology at the process, and eventually process become fragmented, and then they lose the process and become atrophy. And directly, this paper showed the microglia expressing P16 and P21, two direct markers for cellular senescence, are overall increased in a human AD postmortem tissue. And consistent with this finding, the single nuclear RNA sequencing using the human brain, they find that some dystrophic like microglia and also senescence microglia as a subcluster of microglia. So we ask whether autophagy deficiency can cause the microglial senescence. To test this, we ran the single-cell RNA sequencing using the ATG7 condition knockout microglia and then wild-type control. And among the many different clusters, we focus on the cluster that predominantly derived from the ATG7 condition knockout. And interestingly, the cluster 9 contained the increased genes of P21, which is cell cycle inhibitor, and also they highly expressed some chemokines such as CCL6 and CCL9. We also validate the increase of P21 by immunostaining. You can see here. We also performed the cell proliferation assay using EDU, which is a thymidine analog. And we found that the EDU positive microglia portion was much lower in the ATG7 condition knockout microglia. We also measured the volume of a lipofuscin, which is known as autofluorescence aggregate, because those structures are known to be increased with aging. And we found that the lipofuscin volume overall increased in an autophagy deficient microglia. And given that dystrophic microglia shows some like dystrophic morphology, like fragmented process, so we also analyzed the morphology of microglia, and we found that the branch length and branch terminal point both reduced, indicative of a compromised branch complexity in an autophagy deficient microglia. Also, we found that the ATG7 condition knockout brain contained the more microglia that resembling the human dystrophic microglia morphology compared to the control. So also we validate the cellular senescence marker in a cultured microglia condition. And after culturing the ATG7 and ATG14 condition knockout, we found that the lower number of microglia compared to control. And we also find out the increase of a P21 in this two different autophagy deficient microglia model. And also we found that the senescence-associated beta-galactosidase activity also overall increased in this autophagy deficient microglia. So overall, this data indicate the loss of autophage causes cellular senescence phenotype in microglia. We also chased microglia, senescent microglia, by labeling the S100A4, which was the one of the specific marker for cluster 9. we used the RNA-scope in situ hybridization assay. And consistent with this single cell data, single cell sequencing data, we found that the S184 positive microglia which accumulated in the absence of autophagy. And importantly, in a 5x fat background, we found that the S184 microglia lose the interaction with the amyloid plaque compared to this class 7A DAM microglia. So these data indicate the senescent microglia are less associated with the amyloid plaque. Lastly, we asked the impact of a senescent microglia in our study. So we utilized the senolytic drug, which is a dasatinib and quercetin combination. It's a well-known drug combination that selectively removes the senescent cell. So in the autophagy deficient ATG7 condition knockout 5x fat mice, we treated the 11 weeks of a DQ, and we observed after 11 of DQ treatment. And we found out that DQ treatment lowered the P62 containing microglia. So we used the P6 accumulation of a autophagy deficiency, and this data indicate the P6 positive microglia become target of DQ. In another word, a portion of autophagy deficient microglia was indeed senescent. And interestingly, chronic DQ treatment lowered the amyloid plaque number. And we found that the DQ treatment rescued the number of microglia around the amyloid plaque. And also it significantly increased the CLAC7A, rescued the CLAC7A staining intensity. And importantly, we found the DQ treatment convert this diffuse and spiky style amyloid plaque into more compact and round style, as indicated by the increase of X34 circularity. And it was associated with the reduction of a LAMP1 positive dystrophic neurine marker. So this data showed the removal of senescent microglia reverses amyloid plaque associated phenotype that was in 87 condition knockout 5x fat mice. So this will be the second summary. So we found out the resting microglia highly increased the autophagy activity during conversion into the DAM microglia. And as I introduced, the DAM microglia tightly associated with amyloid plaque and minimize the neurotoxicity. But in the absence of autophagy, the microglia becomes senescent. So we termed the senescent microglia as a senescence-associated microglia, SAM. And we characterized the senescent microglia phenotype. And we found that the SAM microglia failed to associate with the amyloid plaque, probably due to the lack of proliferation. And this leads to the increased neurotoxicity. So from this study, we found that autophagy is activated in DAM population, and loss of the autophagy microglia causes a cellular senescence phenotype and aggravate the amyloid plaque associated phenotype. So since two works was done in my previous mentor's lab, Jenny Yue, so like I really thankful my previous mentor, and he support a lot like during my transition, and yeah, at the farewell party, like I ended up crying, like, and it was quite emotional at the day. And also the computational analysis was done by like Bin Zhang's group in the Mount Sinai, and Ming Wei helped a lot. And these works are primarily done by my mentor's NIH R1 grant. And I also used a little bit of like REC award from ADRC that I obtained from Mount Sinai Center. So I will just briefly introduce the ongoing works. So when we think about the AD and PD, two most common neurodegenerative diseases, we will firstly think about aging. So with aging, there's increase of a cellular stress, including the DNA damage, oxidative stress, and reduced proteostasis, such as a reduction of autophagy activity. And literature has shown that this type of stress can cause cellular senescence. So it is expected to see the accumulation of a senescent brain cell with aging. I'm particularly interested in the senescent microglia. And senescent microglia may impair the homeostatic function and increase the secretion of SASP component that can cause the chronic inflammation. So my two previous study support this hypothesis. So like even though I didn't investigate the cellular senescence angle in my first study, I think the lack of alpha-synuclein degradation could be the one of the phenotype of senescent microglia because I used the exactly same animal model. And also from the second study, I directly showed how autophagy division senescent microglia impact the amyloid plaque pathology. So why I'm interested in senescent microglia and why I'm thinking that this cell type is important for understanding the the human brain. So literature, like two different literature showed the microglia are highly susceptible to cellular senescence with aging. So they used like senescence reporter mice line and they aged them and they ran the single cell RNA sequencing. And they found that the majority of the senescence positive cell was actually the microglia. So this like evidence support the microglia, probably the most susceptible cell type for cellular senescence in aging. And also, I can say that there's like two papers show the comparable like microglial turnover rate between human and mice. So this Nature Neuroscience paper showed like about the 26 percentage of microglia turnover every years, and it's very depending on the like a brain region. And also the second paper showed the directly measured microglia turnover, and they found that about 30% microglia per year they proliferate. So which means is likely in the human brain, the microglia will encounter the replicative senescence stress because we live much longer than mice. And third rationale, it could be because human brain contains the greater white matter volume compared to the mice. So as we know, we have a much white matter, we have a greater white matter volume compared to the mouse. And interestingly, also this paper shows, and also I validated in my hands, I found out the microglia, senescent microglia, are highly enriched in the white matter tract. So by three different rationale, I think the senescent microglia probably the very important cell type, subtype of microglia that is important for also understanding the age-dependent and neurodegenerative disease. So consistent with this kind of a hypothetical, like a rationale, is a human brain and in a single cell data, like constantly detect this senescence or dystrophic microglia type in the human brain, which is a very like uncommon in the mouse model. And also many papers already have shown the pathogenic roles of a senescent like senescent brain cell in not only Alzheimer's disease but also in a PD. So microglia or the like oligodendrocyte precursor cell and astrocyte and many paper already have shown the pathogenic role of senescent microglia, but their underlying mechanism is totally unknown. So my overarching goal will be the determining roles of senescent microglia in AD and PD context. So if I just briefly introduce my K1 work, like my previous mentor and our lab, through collaboration, we investigate the like PD transcriptomic change using the single nuclear RNA sequencing. And I participate this project as the validation experiment. And interestingly, in a microglia cluster, we found that some senescence-related genes are increased. So like I wrote about this kind of a proposal as a K1 grant. So my hypothesis is the microglia become senescent, and senescent microglia release some SASP factor, and it can affect the dopamine neuron integrity. So as a first step, I I checked whether there's a senescent microglia accumulated in PD brain. So we get the human postmortem PD brain from Miami NIH Brain Bank, and we found out in P21 and P16, like two different marker for cellular senescence, we found out that this P21 or P16 positive microglia are more accumulated in a PD brain. So I will investigate the like underlying mechanism, like and also validate their function using animal model. So I just made my web page for my lab, and I generate this image through ChatGPT, as you can see, and my lab is aimed to investigate like three different main we'll have three different main questions. So we will determine the how disease condition promote the microglia senescence, and we aim to identify the like molecular underlying mechanism that links senescent microglia to the neurotoxicity. And also, it will be very interesting to see the how ADPD risk factor can affect this microglia senescence pathway that eventually affect the neurodegeneration. So when we see the withering tree in our garden, we will check the water, soil, lighting. So I believe that this is the same as true for understanding the neurodegenerative disease. So neurons are the major cell type that like for the brain functioning, but the non-neuronal cell, astrocyte, oligodendrocyte, and microglia, other glial cell, tightly associated with the neuron and support the neuronal function and survival. So my central hypothesis, compromise the glial function can promote the neurodegeneration. And as a new start, I want to test my first hypothesis, which is senescent microglia may promote neurodegeneration. Thank you for listening.